Gopal, we can wait till 4:10 and then start.
Cool,
Is
cool.
it
That's
fine?
perfect. Yeah, that's fine. That should be fine. Yeah. Thank you.
I think we can start.
Cool. Yeah.
Okay. Hi all. Uh, Welcome to the guest lecture by Dr. Gopal Murali as a part of week 10 lecture series. And before we begin, I will give a brief introduction about Dr. Gopal Murali. Dr. Gopal did his PhD from ISA Trivandrum and completed 2 postdoc, one from Ben Gurion University in Israel and another as a Fulbright Kalam postdoc from University of Arizona, US. He's interested in many areas of ecology and evolution, and his recent research lies at the intersection of macroecology, macroevolution, and biodiversity science. The unifying theme of his research is to understand how ecological and evolutionary processes shape diversity across species, space, and time, and how human activities are altering these fundamental dynamics. He integrates large-scale biodiversity data, advanced modeling, and experiments across diverse taxa to address key questions on trait evolution, species distributions, and extinction risks from a global perspective. I hope all the students will uh, use this opportunity to interact with him, and you can, I think you can stop anywhere in between and shoot questions with him, shoot questions, or type that in the chat box. Over to you, Gopal.
So thank you, Prabita, for the really nice introduction. So can you hear me well first?
Yeah.
Good. That's perfect. So first of all, I would like to begin by thanking for the opportunity to present my work, and it's really nice to meet all the students. And you're having a really amazing time with this fundamentals in ecology NPTEL course. So as a part of this live session today, I'll be talking to you about the work that I did during my postdoc as a case study in macroecology. So before diving into that, what I thought I would do today is to basically uh, go through a little bit of introduction or something like a recap of what macroecology is and the philosophy behind classical ecology and the macroecology. Just to give you a brief recap of what Professor Karthik Shankar has already dealt with in his macroecology module. And as Prabita said, I want this session to be much more interactive, so we can be much casual in that sense, I think. So without any delay, I'll start. So I would like to begin by asking you guys a question. So the question is, can you guess the spatial resolution, for example, the plot size at which most ecological studies are conducted? Or in other words, if you were to do, for example, a literature survey, wherein you just do a Google Scholar search, try to find many different ecological studies and trying to get the plot size of each of these studies. And if you were to draw a histogram of this distribution of plot size, what do you think would be the average plot size or the spatial resolution of these studies would be so far? So I've given you to help you. I've given you a scale here. So it can be less than 0.1 centimeter square, or it can be 1 meter square, 100 meter square, 1,000 meter square, and for example, more than 10,000 hectares. Can you either type in the chat box or yeah, it's better you can type in the chat box, I think.
Students, you can type anything. Any answer is fine. He will correct.
Yeah, it's just a
Gopal will
guess, right?
correct
You can just
easily.
type.
So
Yeah.
can consider it as a very casual discussion. So you can type any answer you think. Gopal, it'd be better if we can also see you.
Oh, you can't see me?
Yeah. Sorry. It's my bad. Yeah.
Okay. So no, so what I'm asking is just an average. If you were to draw a distribution of the spatial resolution, the plot size, what would be the size? For example, the plot, for example, usually we do plot as in as a square grid, right? What would be the resolution of it? For example, 1 by 1 meter plot, or for example, 10 by 10 meter plot, and so on and so forth, right? So it can be any value on these examples that I gave you on these axes, right?
Gopal,
Just take a
there
wild
is
guess.
message, like there is a chat in the chat
Cool.
box.
Yeah,
Can you
yeah,
please
yeah. I
respond?
just saw that. So one answer is just 10 meters square. Is one guess is 10 meters square. So maybe rather than waiting, I'll just show you what actually people have found. So okay, let me change this. So this is what actually done by a study conducted by Estes et al., published in the journal Nature Ecology and Evolution in 2018. What the study is basically done is that they looked at the spatial scale or the resolution of each ecological study, basically from paper published in the year 2004 to 2014, focusing mainly on 30 major journals in ecology, right? And they plotted this resolution of this plot size from each study, right? So what they found out is what you're looking at in this screen, right? So some of you guessed 10 meters square. That's actually much farther higher than the average, right? So from this histogram, what you can see is that the average resolution is about closely between 0.1 meter square to 1 meter square, right? Or to be more precise, what they found is that about two-thirds of the papers that they studied have a plot size of 1 meter square or lower, right? And this is one dimension of the spatial scale, right? So there is also another component, that is the extent, basically how much geographic span that their study basically encompassed, right? And that is the graph that you can see, which is called the extent on your left side. And here you can see that average is about 10,000 hectares, right? And more than half a study they found have the spatial extent of less than 10,000 hectares, right? So so why is this important? So 10,000 hectares is something, if you want to imagine how big the 10,000 hectare size would be, if you were to consider the average, consider the area, urban built area of Bangalore, it just it's just a 5% of that area is basically 10,000 hectares. You can imagine how small these studies were, right? So this sort of highlight that these spatial scales are far below those on which the major anthropogenic influences, for example, climate change and land use, act on, right? So sort of highlight the takeaway point from this slide is that many of the ecological studies are restricted in spatial and spatial resolution. So why is that a big problem, right? So if you were very limited in understanding ecological questions or problem in a very limited spatial scales, then how can we understand how nature works if we study one population or a community or one experimental cell? So the thing is, the experiments that people have been doing in classical ecology, or the experiments basically, are restricted in either time, space, or phylogenetic scope. Of course, this is sort of comes from the limitation of how much work one can do in a span of a human lifetime. For example, if you were to do some ecological studies during course of your PhD, you obviously cannot do studies which are other than, for example, 5 to 7 years. So there are certain limitations of classical ecological studies, but it's also highlight that if you want were to generalize any question or pattern, we are going to a really tricky place wherein your experimental scale in terms of space or time are very limited, right? So in order to illustrate this, I just thought I would just put up this blind man and the elephant story, which has its origin in India, right? I hope some of you would have come across it. So the story goes something like this. So a bunch of blind men, obviously they can't see, are trying to guess how a lizard, how a elephant would look like, right? So what they are doing is basically try to touch different parts of the elephant and trying to guess how an elephant would look, right? One of them touched the trunk, for example, and then he said, okay, elephant looks something like a snake because it's long and tubular. And the other one hugged um, the the leg of an elephant, and he says, okay, something looks like a tree. Another one, for example, touched the body of an elephant, and then he says it looks like something like a wall, right? So our class, we can draw some analogy, this example or story to that of classical ecology, wherein we are in classical ecology, people were trying to understand a problem by breaking them into small parts and guess what would be the major or trying to make a major generalization of that problem. So so just to put things in perspective, to make it very clearer of how classical ecology differs from macroecology, as I said, classical ecology employs a reductionist perspective, wherein it assumes that if you take a big problem, you break them up into small parts, and you try to understand the major picture by using what the knowledge that you gain from understanding this smaller problem. So one example could be that in classical ecology, you can think of is how why particular pollinators are attracted to one specific colors of flowers, right? So one can study this question from different aspects. For example, you can look at the molecular aspects of how colors are perceived by the pollinators, and also one can also look at why how environment, for example, lighting can affect this choice, right? So by doing so, what we are trying to basically do is splitting up problems into really small chunks and then gaining very mechanistic detail. In this case, for example, understanding molecular underpinnings of how a bee would see a particular color based on, for example, the visual pigments in their retina. But as I said, this approach may not be useful all the time. So one classical example can be a quote from Douglas Adams in his Hitchhiker Guide to Galaxy, right? So it says, if you were to try and take a cat apart and see how it looks, how it works, the first thing you will have on your hand is a non-working cat, right? So the underlying point of this is that whether it's a cat or an ecosystem, if you're trying to dissect something to understand it, can sometimes destroy the very thing that you're trying to study, right? So that's where the macroecology comes in, right? So what it tries to do is basically macroecology try to take a holistic perspective, and it's based on the philosophy of holism, which is basically that it says you cannot reduce the parts and retain the meaning of the whole, right? And it's basically try to understand emergent or spatial patterns at a really macro scale when you step back and study the patterns as they are without doing any reductionist approach. So to be very clear, there are I now move on to giving an actual definition of macroecology. There are two different views actually, which has been quite frequently used and interchangeably used sometimes. The first definition is that macroecology is just ecology at large scale, as you can guess from its name, which is basically the study of ecological patterns and processes. It can have three different dimensions. One is the scale of space, or the scale of time, or the scale of taxonomy, or the taxonomy. So there are no hard rule to say, okay, once I cross the threshold, the number of species, I'm going to call that study as macroecology. So Brian McGill in his recent paper trying to frame how one can call a macroecological study based on these different axes. So anything in hundreds or thousands, he says, he recommends basically anything in scales of hundreds or thousand, one can safely call it a macroecological study, right? For example, spatial scales of thousands of kilometers square, time of thousands of years, or species having hundreds or even thousands of species. And if you have more than a large dimension in any one of these axes, you can safely call that study as a macroecology. And the second definition is that it takes a little bit of different perspective. It focuses more on the emergent ecological patterns. It goes like macroecology is something like a study of ecological pattern that arise from collective behavior of organisms, shaping biodiversity and ecosystem beyond the sum of its parts. So the emergence here refers to the patterns or property that arise from interaction of individual components, but cannot be predicted simply by studying those components in isolation, like what we saw in classical holistic perspective, where things won't work if you break the system apart. So there are classical example of this, as Professor Karthik Shankar has already covered many of those in his previous lectures. One is the latitudinal diversity gradient, which is sort of an emergent property which you can only study at macroecological scale. So with that introduction, I'll now move on to the case study that I'm going to discuss today. So the title of this talk is going to be like the origin and future of evolutionary and geographic rarity in terrestrial vertebrates. So this study basically tried to understand why certain species are evolutionarily unique and also geographically restricted, and also basically trying to map where these species are found, trying to understand what are the determinants of these hotspots of species, regions containing these species, trying to understand if there are any environmental factors that make species only restricted to a particular region and evolutionarily unique, right? And also at the end, I also did some analyses to try to look at the conservation implication of this, which I'm going to talk about in this talk today. So for those of you who want to take a read of this study later on, if I have missed something, you think I did not explain things clearly, you want to read it later on. This paper has been already published in 2021 in the journal Science Advances, which you can check later on if you need. So to begin with, one of the major macroecological pattern that we see in nature is commonness of rarity. So what do I mean by that? Some species are quite widespread and they're found like all over the world, but many more species are rare. So these rare species, so this is true in terms of scales of both abundance and spatial scale. So what today I'm going to focus only on the spatial scale, which is like distributional range. Some species are found only in some part of the world, for example, the Western Ghats. Some are found, for example, throughout the world, right? For example, the foxes. So the pattern that we find in nature is that many species are rare, or in other words, many species are endemic. So just to give you an idea of what do I mean by that is that if you were to plot the geographic range size of species, which is basically a distributional range, as a QQ plot, what would you get is something like this. So this plot is shown for reptiles, right? Geographical range size. And what you see is that there is obviously a lot of deviation at the lower and higher value. So, what does this mean is that because there is a deviation from this diagonal line, there are many more small ranged species of reptiles, and there are quite a few large range species, but there are very large in terms of their distributional range than predicted from normal distribution. Or in other words, if you were to plot the geographic range size uh, of a species, of all the reptile species as an histogram, what would you find is that the histogram is basically right skewed, meaning there are many small species, and also has a long tail. Long tail meaning there are few species which are completely widespread and found all over the world. So this question of why there is many rare species has troubled ecologists and evolutionary biologists for a very long time. And it also takes a central place in conservation because these geographically rare species are also highly prone to extinction. So obviously a question arises when you look at these distribution patterns of species is that what makes some species and clade spread geographically while other remain confined in space over geographical time? Or in other words, if you were to plot the distribution of these small range species, what would you obviously see is that these species are not like scattered all over the world, but obviously they are present only in some part of the world, what we classically call the hotspots, right? But but people have so far studied looking at only the species distribution, but there is also some component of geological time involved in it, right? So this thought of correlation between geological time and species distribution is not like something that people have been thinking seriously in the recent time. And it dates back to even Darwin, right? So Darwin, in his book, wrote that I think it inevitably follows that as new species in course of time are formed through natural selection, others will become rarer and rarer and finally extinct, right? So Darwin sort of trying to understand why, trying to answer distribution of species in terms of also the time component. What he is trying to say is basically, as the new species are formed, they are probably restricted in geographical space. And it's also equally true for species which are old because they are in the process of getting extinct and therefore they're being outcompeted by many species in their geographical range. So this sort of highlights some evolutionary time component in how we need to think about these rare species, right? So people have actually come up with the idea of combining endemism, or in other words, spatial restriction in geographic ranges of species and the evolutionary history. And there have been two schools of classification of what one can be considered, especially in view of, for example, what Darwin has been saying in terms of origin versus extinction of species, right? The first idea is neo-endemism hotspot. So these are basically regions or hotspot areas that include species that have been originated very recently and they are yet to expand. Therefore, they are basically spatially restricted and leave only those regions where they just recently originated, right? So these are basically are also otherwise called as the cradles of diversity, basically where species are it's like a speciation center where species are basically speciating. So one classical example that one can think of in Western Ghats is the southern Western Ghats, especially a group of tree frogs and bush frogs, right? Philotis and Rochesters. These frogs have incredibly higher amount of diversity because they're restricted to highly topographic complex region, basically mountain regions that potentially have higher speciation rate. And the second view is paleo-endemism hotspots, which are considered also the museums of diversity, which are basically hotspots or which include regions that hold species that have diverged quite a long while ago, but then they're present only in those regions because they have never expanded over their range over time, or it could also be that it was once widespread, but then currently, as we see, their ranges have contracted, right? They're once widespread, but then in many parts of their distribution, they're gone extinct. And right now, they're restricted only to certain parts of their distribution. So one classical example of these species could be our purple frog, again, that is found in the Western Ghats, which is the Nasica batrachus frog. As you can see, you don't have any recent relative of this species found in India. The only closest relatives are found in Madagascar, right? So this is the only, again, this is only the only species found in this particular family, right? So again, it's quite restricted to in its distribution only in Western Ghats. So having given that background information, I'll move on with the objective of what exactly I did in this particular study. So we posed a bunch of questions with regard to species distribution, especially endemics and their evolutionary history, right? So the major question we asked is to basically plot where these evolutionarily unique or evolutionary rare and narrow range species are located, basically identifying the hotspots or regions across the world where you can see many uh, uh, species that are quite restricted in geography, meaning they have small ranges and also evolutionarily unique. So we did this study across all tetrapod species, land vertebrates, or in other words, which are basically amphibians, birds, mammals, and reptiles, right? So the second question we asked is that how congruent are these regions for different classes of terrestrial vertebrates? Meaning, does different groups of classes of vertebrates show different hotspots. For example, we know from earlier studies that reptiles love desert or hot region. So therefore, for example, these evolutionarily unique narrow-ranged hotspots are found, for example, in desert regions, like for example in Australia. And it could be, for example, different for other groups as well. For instance, amphibians could have, for example, hotspot in the Amazonia, right? So basically, we were trying to understand in the second question how congruent or spatially overlapping these hotspots are for different groups of land vertebrates, right? So this has got important conservation implication, because if we were to conserve these species, if these hotspots are overlapping for all the four groups, then our job is easier because we just have to protect those regions. But their hotspots are not spatially congruent, meaning different species have different hotspots in different parts of the world. Then it means I need to conserve, we need to pay unique attention to different uh, regions in the world in terms of conservation. So we also, the next question is also identify hotspots of new and paleo-endemism, which I already explained in the previous slide. Basically trying to understand whether there are certain hotspots which are like cradle where the new species are originating, or the hotspots are basically museums where there are really evolutionarily old species. They're just persisting there because of many other ecological reasons that I'm going to explain you in a couple of other few slides to follow. And we basically also followed, as a follow-up question, we want to understand what are the main determinants of these paleo and neo-endemism hotspots. And as I said, because these important conservation implication, we also tested how well these hotspots of evolutionarily unique narrow range species are protected, and also looked at what are the anthropogenic threats, for example, land use changes, climate change, and for example, bunch of other threats are there in this particular hotspot, and also the species that contribute to these hotspots, right? So these are the major objectives of our study. So in the following slide, I'll move on to explain focusing on the third or the fourth objective. What are the determinants of these endemism hotspots? So the first hypothesis that we came up with is basically the climatic stability hypothesis, which was calculated as a velocity of climate change since the last glacial maximum, right? So this hypothesis is related to persistence of species in that hotspot, and therefore it's related to phylogenetic endemism. So the idea is that areas with low velocity of climate change since the last glacial maximum acts as a refugium for these small range species. So this pattern can arise because of two different reasons, right? So on a long term, less stable climate selects for species with high dispersal ability and therefore reducing endemism, right? On the contrary, when you have highly stable climate, they're not restricted to disperse out. Or it could also be an entirely different hypothesis, is that species with small ranges in climatically unstable regions may face higher extinction risk. Therefore, one would expect stable climatic condition to be correlated with these hotspots of paleo-endemism, right? Um, this has been quite studied well previously, and people have shown that these hotspots of species endemism tightly correlate with climate change velocity since, for example, the last glacial maximum, right? So I should acknowledge here that long-term climatic stability can mean different thing for many species. It's just not that climate change since the last glacial maximum. It can go back in very deep time, but we could not test this hypothesis at deep time because we don't have spatially resolved data uh, beyond LGM. So therefore, we restricted our analysis till last glacial maximum, right? So the second hypothesis we tested is the climatic rarity hypothesis. So what this says is basically regions with distinct climatic condition than those of surrounding areas select for local adaptation, or in other words, specialization, and therefore limit range expansion. Or in simple terms, so the current region where, for example, a species is there, it's quite unique when compared to the region surrounding it, right? So therefore, once the species start specializing to that particular location, they cannot move out because the regions surrounding them are completely different. Therefore, they cannot survive, right? So therefore, those regions with such climatic unique conditions promote local phylogenetic endemism. So this has already been shown. For example, for birds, I think this one particular study. And moving on, we also have came up with a bunch of hypotheses for the determinants of neo-endemism, right? So here, one of the idea is that heterogeneity in terms of habitat. So heterogeneity can mean in many different axes. For example, the differences in soil types, or for example, elevational range. So the major idea behind heterogeneity promoting endemism is that in heterogeneous or topographically complex area generates dispersal barriers, therefore that promotes specialization, creating small range species, right? In our study, we use three different metric of heterogeneity, which is the soil type present in that number of soil type, distinct soil type present in that particular location, or the number of ecoregion, or for example, the elevation range, right? Again, this hypothesis is related to origin of endemics, right? Because it sort of promotes, indirectly promotes speciation in that particular location. And the second idea or hypothesis is that productivity, this is again related to origin of species. The idea is basically that high energy availability may increase specialization and therefore opportunity for speciation, promoting endemism. And the final hypothesis deals with current climatic seasonality or climatic variability hypothesis. I remembered Professor Shankar have touched on this aspect in his previous lecture. This is also known as related to Rappaport rule. Uh, can maybe any of you explain what the Rappaport rule is? You can type in the chat box what basically Rappaport rule is. Okay, maybe I'll just go and explaining it. It seems like you guys are shy. So the climatic seasonality or climatic variability hypothesis, or on other terms related to the Rappaport rule, basically says that regions with less stable climate, or in other words, seasonal variation in climatic conditions selects for species with broader climatic tolerance and therefore larger ranges, right? So what do you mean by seasonal variation is basically species that are found in higher latitude sort of experiences quite a lot of seasonal fluctuation in temperatures that they encounter, hot and cold condition in summer and winter respectively. And therefore, when they encounter that, they evolve broader climatic tolerances and therefore they tend to have larger ranges, right? So that's the basic idea behind the climatic seasonality or climatic variability hypothesis. So with that brief idea of the prediction that we tested on why certain species are evolutionarily unique and narrow ranged are restricted to certain parts of the world, I'll now move on to the methods. So for this particular study or any macroecological study, what people normally use is to take distribution data in terms of species range map, for example, the IUCN. We also did the same. We took range map from the IUCN for 30,000 terrestrial vertebrates, which includes, again, birds, mammals, amphibians, and reptiles, which encompassed about 84% of all known species. And then we converted them to a presence-absence matrix using an equal area grid at a resolution of 1 degree or 100 by 100 kilometer, right? So we used 1 degree resolution because this is the data that this is a limitation of the data. IUCN data are not really fine grained. They're not meant to represent species distribution at a really fine resolution. So therefore, we restricted to our analysis to 1 degree resolution of scale. Basically, the grid that you see in in the subsequent following slide is basically has a height and width of 100 kilometers. And we used molecular phylogeny from recent studies, and we used about 100 trees for each group just to account for phylogenetic uncertainty. And for climatic and other data, we used data from the CHELSA dataset for current climatic data, included all bioclimatic variables from 1 to 19, and also bioclimatic variables since the last glacial maximum. And for elevation, we used SRTM data. And for soil type and ecosystem per grid, we used already existing data, and same for net primary productivity. So rather than directly jumping into the results, I thought I would briefly go over how one can calculate phylogenetic endemism, right? So phylogenetic endemism is a metric that is actually used in our analysis to quantify uh, the regions with that hold evolutionarily unique and narrow range species. So this is the actual metrics that quantifies whether a region contain highly evolutionarily unique and narrow range species, right? So I'm going to interchangeably use this term phylogenetic endemism in the subsequent slide. So which is calculated basically as the sum of branch length connecting a species or a descendant from a one particular community or a grid cell or assemblage divided by the number of grid cells where a species or a descendant species occurs. So it might be quite tricky to understand just from this definition. So what I thought I would do today is just briefly go over of how this is calculated. So imagine you have three species A, B, and C. A is found only in this corner grid cell, B is found in these two grid cell, and C is found in these two grid cell, right? And they have phylogeny something like this. B and C are closely related. A is quite ancestor to B and C, right? And you have ancestor clade D, which branch up to C and B. And the values that you see are the branch lengths, right? So given these occurrence matrix and phylogeny, how do you calculate phylogenetic endemism is basically like this, right? So for A, it's quite simple. You just take the branch length, which is 2, divided by the distribution that particular clade occupied. Here, it's 1, right? It's just found only in one particular grid cell. So you divide 2 by 1, you get 2, right? And things become a little tricky when you start calculating the other grids, because for this particular grid cell, you have the region sat by two different species. And this particular grid cell, although you just have only one species there, it is found elsewhere also. So how do you calculate that using this formula? So for C that is found in this particular grid cell, what you do is basically take the branch length of that particular lineage 1.5 and divide that by the number of grid cell that is present, right? 1.5 divided by 2, which gives 0.75. But then you also have these ancestor that you need to account for. The branch length, which turns out to be 0.5, and you have this particular clade occupied by species C and B, which is found in three different grid cells, right? A, B is found here, C is also found here, and C is also found here, right? So you divide this 0.5 divided by 3, which you get 0.176. So if you combine this 0.75 plus 0.167, you get 0.92, right? Likewise, if you were to calculate that for this particular grid cell for B, you would get exactly the same value, because here 1.5 divided by 2, you get 0.75. Again, you have a ancestor clade, which is 0.5. Again, you divide that by 3, you get 0.16, again, which is equal to 0.92. So for the other grid cell where you have both species B and C, what you do is basically repeat the same, but separately for C, B, and D, right? So for C and B, we know 1.5 divided by 2, 0.75 and 0.75. For D, again, if you divide this value by 3, you get 0.1675. And you sum it up, you get 1.67, right? So I went through this process to demonstrate that. So what this basically these value now tells us is that this particular region that holds species A, it's quite evolutionarily unique, and obviously it's not found elsewhere in the steady matrix that you have, right? On the other hand, although B and C are found in two different grid cells, they're quite okay in terms because they're found in other regions also. Or more importantly, when you look at the cells that contain both species, it received the second highest score because naturally, when you have many more species and you sum up the branch lengths, you will obviously get higher amount of phylogenetic endemism score, right? Simply because you have many more species there, you sum up the branch lengths, you will get higher value. Again, but still, you are weighing them by range size, and therefore it's lower than 2. Therefore, if one were to say which out of these 4 cells which have higher phylogenetic endemism, is just that cells containing A followed by B and C. So we basically calculated this at the global level for all the grid cells that we have using our distribution and phylogenetic data set. But as I said, this phylogenetic endemism score, it's not like it's quite sensitive to the species richness of each grid cells. So therefore, one need to do some randomization to identify regions that hold disproportionately higher evolutionarily unique and narrow range species. So what can one do is basically do some randomization analysis wherein you can, if you imagine if this were to be the original matrix as I shown in my previous slide, what can one do is basically retain the same number of species for each grid cell, but then you just rearrange them in terms of spatial scale. So after doing this, what can one do is that after doing that for 1,000 times, one can compare this value or randomized value to the observed value. So if the observed value was way higher than the phylogenetic endemism calculated during randomization, for instance, here we used a 5% significance threshold for calling something as significant, and it's a two-tail, therefore you have 975. So if the randomization value 975 times was lower than the observed phylogenetic endemism score, then what it tells us is that that particular grid cell holds highest level of phylogenetic endemism, right? So this is basically to identify hotspots of phylogenetic significant phylogenetic endemism. And one can also do the same thing to in terms of the clade level. So this is basically to identify hotspots of paleo and neo-endemism. What we did is basically take the original phylogeny, which is called P original, and you calculate what's called the relative phylogenetic endemism, which is basically the uh, the ratio of P calculated using the original score versus phylogenetic endemism calculated using phylogeny where all branch lengths are made equal, right? So by doing so, what it tells us is that if P original is greater than P equal, it means that in the observed data, the branch length is quite really long in that particular locality than one would expect based on randomization, right? And therefore, that particular region holds evolutionarily unique or paleo-endemic species, which are like there for a really long evolutionary time, and they have diverged like really long ago. And see, the opposite applies for neo-endemism hotspot, wherein if the P or this ratio is lower, where P origin is lower than P equal, one can call neo-endemism, which means that that region holds species that have just diverged recently, and therefore they contain they're basically termed as the new endemism hotspot. And you can also have a third case where P original and R equal to P equal. This is where what's called the mixed endemism hotspots. Basically, it tells us that these region hold species that are both evolutionarily unique, evolutionarily diverged long ago, and also few clades that are diverged recently, right? It's a combination of both neo and paleoendemism. So so I'm not gonna get in detail with the determinants of phylogenetic endemism because um, I've already sort of explained the hypothesis. So I'm just going to quickly go over this. For climatic stability hypothesis, we calculate the velocity of last glacial maximum, which is basically the rate of temperature change over time since LGM, but divided by the spatial gradient of temperature. So we also did analysis using precipitation here to calculate the climatic stability. So for the second hypothesis, climatic rarity, how we quantified rarity is basically we took all bioclimatic variable 1 to 19, and then we did a PCA on it. And then using those PCAs, we calculated the mean Euclidean distance between the focal cell and the neighboring cells. So if the Euclidean distance is higher, what it would tells us is that the climatic condition of the surrounding cell for that focal cell is very, very higher or very different drastically. And for heterogeneity, as I said, we use the number of soil type for ecoregions. We use the number of ecoregions, and for elevational range, for heterogeneity in terms of elevation, we use maximum minimum of elevation in that particular grid cell, right? Basically, what is the number of what is the lowest elevation in that particular region versus the highest elevation in that particular region. Region is basically the grid cell. So for productivity, use net primary productivity, and for the current climatic seasonality, we used Bio4 and Bio5, which is basically temperature and precipitation seasonality from the Chelsea dataset. So we used a multivariate regression framework to account for spatial autocorrelation. So the major problem or thing that you need to account for in many of the macroecological studies is spatial autocorrelation. So do you guys have an idea of what spatial autocorrelation is? I think Professor Kavita Ishwaran have talked a little bit or talked quite a lot on this spatial autocorrelation, pseudo replicates, and so on and so forth while doing experiments. In classical ecology, you can actually account for spatial autocorrelation because you are designing the regions, are you can potentially to a degree account for spatial autocorrelation um, by designing or choosing the plot which have technically dispersed in space, for instance. But for in macroecology, it's quite difficult to sort of account for it. And rather than what it's classically done is basically perform an analysis which accounts for spatial autocorrelation. So to give an idea of what spatial autocorrelation is, it's somewhat what is called as the first law of geography, which says everything is related to everything else, but near things are more related than distant things, right? So this is proposed by Waldo Tobler, and he's considered as the father of geography. And in a very simple term, what it means is that if one were to plot, for example, elevation map, and if we were to take two different points, for example, this one versus this one in space, these 2 points, their elevation value are not completely different from each other, right? Because they are spatially, points are spatially closely related to each other. They are obviously found in mountains, and therefore there is a strong correlation between those values that you get in a nearby localities, right? So if these elevation map or the elevation values are actually found to be random, this is how things would look like, right? So in many variables that we take in macroecology, for example, temperature, precipitation, and many other things like elevation here, they are all the time or most of the cases are spatially correlated. In a sense, locality B, value from locality B, for example, here versus point from locality A, it's not completely dependent. You cannot say these 2 points are independent. You cannot treat them as an independent points. But what one becomes is that they are quite sort of a pseudo replicates, right? So we calculate this spatial autocorrelation using what's called the Moran's eye. So this value range from -1 to 1. When you have a value of 1 positive value, it means there is a strong spatial autocorrelation. When you have negative value, then it means that there is a strong dispersion, right? When you have a value of 0, it means they are randomly distributed. And there are also many other statistical analyses that sort of accounts for spatial autocorrelation. I'm not going to go into details of that, but in our analysis, we sort of perform those regressions, which accounts for spatial autocorrelation. So with that, I'll move on to the results of what we found in this particular study. So the
Gopal,
graph that you're looking
can
at,
I?
yes, please.
We might need to stop in 10 minutes. So,
Okay, cool. Cool.
yeah.
Yeah, there isn't much. I'm going to quite rush over these results. So what we find is this study to answer objective one is that these are the hotspots of phylogenetic endemism. As you can see, these are quite strongly correlated with richness, and therefore you see a mid peak at close to latitude 0. But once after accounting for richness, what you see is that the phylogenetic endemism or these hotspots are quite high along the coastal areas, right? So this sort of supports the mountain speciation model, which says that these which proposes that these diversification centers in the mountains frequently feed species to the surrounding lowland areas, and therefore you see higher sort of narrow range species right next after the mountains, right? And obviously land area also constrained phylogenetic endemism. As you can see here, that phylogenetic endemism value is quite high in lower hemisphere. That's simply because the land area available at the lower hemisphere is quite high. So we sort of looked at this by calculating the coastline and mountain proximity to each grid cell to phylogenetic endemism. And we obviously find there is a really strong positive correlation. So to address next question about spatial congruence of hotspots across tetrapod class, what we find is that over 9% of these hotspots are shared by all 4 species, all 4 classes, basically birds, mammals, and reptiles. So these black regions, which include Caribbean, the Andes, for example, the Western Ghats, Madagascar, Indonesia. And more importantly, about 50% of these areas are never shared across classes, which means only one of these classes are found in this region. For instance, reptiles are found only in this area. Hotspots of reptile phylogenetic endemism is found only in this area, for instance. For birds, Himalayas is one of the hotspots of phylogenetic endemism, which are not shared by technically other groups. Likewise, that also applies, for example, mammals here. This one particular example is a marsupial mole found in the central Australia. So the major point take-home message is that although all 4 classes share their phylogenetic hotspot in many different regions, about 50% are quite unique. And moving on with the hotspots of phylogenetic paleo and neo-endemism, what we find is that we don't find a clear-cut pattern. Many of the hotspot, about 85%, are quite mixed endemism hotspot, meaning they contain both paleo and neo-endemic species. Although there are few regions that contain neo-endemism hotspot, for example, in Saudi Arabia for birds and in the southern Andes for lizards, for instance. And when we looked at the mechanism that's underlying generation of neo and paleo endemism hotspots, what you see here is the comparison of each of these factors that I explained you previously. If it is blue, it means that neo endemism hotspots have lower value than paleo endemism hotspot. White means they're equal. Red means neo endemism hotspots have higher value than paleo endemism. So while just looking at it, you can see that there is no clear-cut pattern, but there are some cases are in direction that we expected. For example, for temperature velocity, we see that these neo-endemism hotspots have higher value than paleo-endemism, meaning there are, for mammals and reptiles, the paleo-endemism hotspots have long-term, lower long-term temperature velocity, or in other words, they are paleo-endemism hotspots are found in climatically stable regions. The same also applies for climatic uniqueness. You can see that for birds, neo-endemism hotspots have lower climatic uniqueness value compared to paleo-endemism, which means that the regions surrounding paleo-endemism hotspots for birds are quite unique, and therefore they are particularly restricted. So one obvious pattern that we find across these comparisons is that elevation range in most of the cases what way higher for neo-endemism than paleo-endemism hotspot. This sort of tells us that elevation has a stronger, similar consequences of paleo and neo-endemism hotspots for across all land vertebrate classes. So just to look at it from a different perspective, the graph here shows the elevation range for different types of hotspot grid cells. And you can see in most of the, almost in all the cases, the neo-endemism hotspots, which is colored red, have higher elevational range than, for example, paleo-endemism hotspot. So this sort of tells us that these mountain regions sort of acts as a cradle of diversity. So because we are limited in time, I'm just going to quickly go over the other two slides. So the major take-home message for other two things that we also find which is relevant for conservation is that most of these phylogenetic endemism hotspot are less protected, right? They don't have uh, protection compared to, for example, the non-hotspot regions or the 10% threshold that people usually use. And finally, we also found that these hotspot region also face greater exposure to future climate change. So this red colored value shows that velocity of climate change in the future. And one can find that these values are quite high across all the four classes, that the climate change is going to be much higher in these hotspot cell compared to non-hotspot. So with that, I conclude my talk with highlighting few take-home messages, which is phylogenetic endemism is high in coastal areas near mountain system, supports the mountain speciation model. And phylogenetic endemism hotspots are not spatially congruent across taxa, as nearly about 50% of the identified hotspot cells belong to a single class, which is again have important conservation implication because we cannot rely on single taxonomic group as classically done, for example, mammals and birds to prioritize conservation, right? Which means we need to pay unique effort in conserving these separate hotspot for each of these four classes separately. And the mechanism underlying a generation of these new and paleoendemism are idiosyncratic, but then there is a strong importance of mountain uh, as their role in these cradle of diversity in generating these phylogenetic endemism. And finally, these phylogenetic endemism hotspots are less protected and also threatened by future climate change. With that, I'd like to acknowledge my supervisors, Professor Shai Miri and Professor Uri Roll, the lab members, and the funding agencies. And with that, I'd like to take any questions if you have time. Yeah, thank you.
Thank you, Gopal. Students, can you post your questions? We have only 2 minutes left. So can you post your questions or just unmute and ask to Gopal? Gopal, can I ask one question?
Yes, please.
Yeah. So, can you explain why do you why mountains are the cradle for? Is there a
So,
reason why?
so there are
Or
many hypotheses or many explanation. Yeah, so exactly. That's, I think someone also asked the same question. Could you summarize the effect of elevation on neo and paleoendemism, right? So the idea is that these mountain sort of um, acts as a dispersal barriers, right? If we have high topographic heterogeneity, that's sort of species to specialize, that sort of acts as a dispersal barriers, and therefore species specialize to only those particular locations, right? And therefore, when you have that, one species started specializing only to particular elevation, you obviously have a higher amount of speciation, and therefore you have many species that are geographically restricted only to those areas. On the other hand, for paleoendemism, mountains do act sometimes. In paleoendemism hotspot, we also find higher elevation range compared to non-significant grid cell. That is also because in some of the cases, these mountains acts as a refugium for species buffering from long-term climate change. One classical hypothesis, for example, here in India is that these southern Western Ghats acted as a refugia, for example, many species during glaciation event, and therefore we see many endemic species found especially in the southern Western Ghats below Palghat Gap, for example. Yeah.
Thank you, Gopal. So by this, I think we have to end the session.
Yeah.
And if you have any further questions, please post that in the Q&A session, the discussion forum. I can pass to Gopal and I can send his reply, add his reply to the group. So thank you, Gopal, for this interesting talk.
Cool. Perfect. Thank you so much. Yeah. Thank you. Bye.